So hello friends, in the previous classes we are discussing the enzyme properties, mode of enzyme action, enzyme classification. That continue with the factor that will be affecting on the enzyme action. Just we can look at what are the factors that will be affecting on the enzyme activity. The first factor is the enzyme pH. The usually all media that consist of the pH is the hydrogen ion concentration, the how actually it will be affect on the enzymatic reaction. The enzymes are active at the optimum pH and loses their activity at the even the pH as a tertiary structure and this is disrupted. Okay, so usually amino acid with the proper charges have the optimum pH. The graph is showing that the optimum pH, that's almost 7, is the better for the enzymatic reaction. If it is high or if it is low, then definitely enzyme reaction will be decreases. That's why when you put the graph, it will shows the pH versus enzyme action. It will show a complete peak shaped graph. In between, that's almost pH 7 is the better for the enzyme activity. So usually the majority of the enzymes, they will have an optimum activity at 7.4. Certain of the organs that in the low and high pH also. For example, in case of the pepsin in the stomach, the optimum pH is 2. Ureases in liver, that's the pH is 5. And even trypsin, that's in the pH is 8. But isn't better is 7.4 pH is having the high enzymatic activity will be showing. Next is enzyme concentration. If you put the plot a graph, enzyme action versus enzyme concentration, that you will find a straight line graph. That means the rate of reaction increases, the enzyme concentration increases. At the high enzyme concentration, more substrate bind with the enzymes. Next is substrate concentration. The rate of reaction increases with the substrate concentration increases, and the maximum activity occurs when the enzyme is saturated. That's if you put a graph, that's the reaction versus enzyme substrate concentration, you will find the there will be a little bit lagging graph, you can find it out. They are shown in the figure, that's enzyme activity will be, and there will be if the maximum activity occurs when the enzymes will be stable. And the temperature, the optimum temperature, that is usually the 20 to 25 degrees Celsius temperature has shows the maximum activity. If the increase in the temperature, definitely the enzyme activity will be decreases, and the rate of concentration of the end product, that will also affect on the enzymatic activity. Substrate concentration I already told. And next, going to the mechanism of action, the enzyme that catalyzes the reaction. The proper fitting of the substrate and active form is required. Enzyme plus substrate, it will give to the enzyme substrate complex. So E plus S give rise to the enzyme substrate constant. It will be reversible, but ES is given as to E plus P, and it's a irreversible in nature. So this is how actually the picture is showing that the enzyme plus substrate, they will be give the enzyme substrate, they will be irreversible in nature. So this is the mechanism action will shown by the diagram. It's a reversible and the irreversible reaction. So for the explaining this mechanism of action, there is a theory will be there. That's a lock and key model or the lock and key hypothesis. So in this reaction, the active sites have the rigid shape. Only substance with the matching shape can be fit into the site. If you look at just like a figure, the substrate key that fit the lock of the substrate is a key that will fit to the lock. So usually if what you looking in the picture, so the same thing, so complete lock and key hypothesis, just like your what you are using the lock and key for the locking system. There will be a one lock and there will be a one key. The each lock and the key will be how the specific, just blue color picture is showing the lock and red color one is act like a key, that's a substrate. The enzyme is a lock, substrate is a key. So like that, when the particular enzyme bind to the particular substrate, then it will be enzyme substrate complex will be formed. The next is induced fit theory. So here there is an active site that's a flexible but not the rigid. Here the what happens, the shape of the enzyme activate site and substrate adjust to the maximum fit, which improves the catalyst. There is a great range of substrate specificity will be occur. So induced fit model, here there will be a enzyme and the substrate, they will be fit to the enzyme substrate complex will be formed, but here there may be a flexibility is there. But in the key lock and key hypothesis model, they mentioned that there is no that flexibility, they have the just like lock and key mechanism. So this is how actually the picture is showing that the how enzyme catalytic reaction will be takes place. So sucrose bind to the active site, that's a sucrase as an enzyme that involved, and the finally sucrose will be formed. So like that, this is the complete structure. First step in the sucrase catalysis hydrolysis of the sucrose, and the third step the produce the glucose and the fructose will be formed. And the sucrase is there ready to catalyze the catalysis of the hydrolysis of another sucrose. This is how the enzyme catalytic action will be takes place. The one is the first is to enzyme catalytic hypothesis explained by, one is a lock and key model and the induced fit theory. All this explains the enzyme mechanism of reaction. So this is how the different enzyme activity will be takes place, how actually the enzyme mechanism will be takes place. Okay, so this is an overview of the enzymatic mechanism. So in the today's video, so we have discussed the what are the factors which will be affecting on the enzymatic reaction and the mechanism of the enzyme action that we have studied in this video. So totally we have discussed the complete account on the enzymes in the last two videos, that's enzyme classification, enzyme properties, enzyme structure, coenzyme, apoenzyme, holoenzyme, prosthetic group, and the other mechanism of the enzyme action, ICBN of the nomenclature of the enzymes, and the different types of enzyme and the different classification of the enzymes, and the factor affecting on the enzymatic activity, and the last one is the mechanism of enzyme action that you had studied in this chapter. So I hope you will got the all complete explanation about these enzymes. So in the next class we will study the another concept from the physiology. If you have any doubts, so you can clarify or send a message to the clarification. Thank you, Ananda. So these are the steps that's enzyme concentration.